everyone. Welcome to today's webinar, Best Practices in Host Cell Impurity Analytics, using advanced technologies and methods to monitor, control, and quantify host cell proteins. Some objectives that will be covered in today's webcast are a brief history and advances made in HCP analytics, HCP ELISA selection and how to ensure the selected assay is appropriate, and advanced methods for HCP antibody coverage analysis, HCP identification, and quantification in drug substance samples. I'm Megan Manzano, the Custom Content Specialist for BioPharm International, and I'll be your moderator for today's event. We are pleased to bring you this webcast presented by BioPharm International and sponsored by Cygnus Technologies. I would like to share a statement from our sponsor before we begin. Cygnus Technologies, part of Maravai Life Sciences, is the biopharmaceutical industry's partner in analytics for host cell proteins, other process-related impurities, and viral clearance. Cygnus helps companies developing therapeutic proteins, vaccines, antibodies, plasma derivatives, and gene therapies to ensure the safety of biotherapeutics prior to human trials, regulatory approval, and commercial release. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found at the bottom of the video player. You can enlarge the slide window by clicking on the small icon in the bottom right corner of the media player. The slides will advance automatically during the event. If you have any technical problems viewing or hearing this presentation, please click on the question mark help widget in the top right of your presentation window. I would now like to introduce today's speaker for today, Eric Bishop. Eric Bishop is the Vice President for Research and Development at Cygnus Technologies. Eric leads the research and development team and is responsible for new product development and custom development services. Eric has over 25 years of experience in HCP analytics. Prior to joining Cygnus Technologies in 2010, Eric worked for AstraZeneca, formerly MedImmune, from 2002 to 2010. During his time there, he had multiple roles from HCP assay development and tech transfer to being analytical representative on CMC teams, guiding projects from IND to BLA submissions. Thank you so much, Eric, for joining us today. I'll hand the floor over to you to get us started.
Great. Thank you for that introduction. Again, my name is Eric Bishop, Vice President of Research and Development here at Cygnus. First of all, thank you all for taking the time out of your day to join us. Hopefully this will be informative and we can have some good questions at the end. So just a quick outline of today's conversation. I really want to go through a brief history of host cell protein analytics. I feel it's pretty important to understand where we started, to understand the journey that we've taken and where we are today. Some of the advances in analytics that have helped us make that journey, how that you can make sure you're using the best host cell protein ELISA possible, and some of the tools that you can use to answer those questions. And then we'll talk quite a bit about our antibody affinity extraction for coverage analysis, but as well as for identification and quantification of HCP and drug substance. So first, as like I said before, it's really important to understand kind of where we started. As she mentioned at the introduction, I've been doing this for a very long time. My first foray into host cell proteins was actually in 1998. So I've been doing this for about 27 years. At the time, there really wasn't a lot of guidance, if any guidance, as far as to how you monitor host cell proteins, because they really were not seen as a potential safety issue. It was more of a process monitoring tool. So you could kind of monitor the purification process each time you ran it and look at the clearance of host cell protein to see if your process was performing optimally. And if you could see differences, then that was kind of a flag to investigate. So as such, there really was not a lot of, like I said, guidance. So there was a lot of ideas and a lot of different methods that were being used. These include Western blots, the host cell protein ELISA that we use today, and even SDS page. Starting in about 1999, we had our first stab at some kind of regulatory guidance, which was Q6B. That was putting some guidance around product, but also the first around residuals. There was a couple important things that came out here. One was kind of highlighting, again, where we were as an industry in 1999. With monitoring host cell protein, it was essentially do the best you can. take every precaution that you possibly can. But also, if you feel like you've done a great job with your purification process, you may not even need to set specifications for host cell proteins. But I think what I really bring from that document, what is probably the most important piece of information, is it did acknowledge throughout the document that host cell protein and biopharma in general was kind of at its infancy. And they understood the methods that we were using at the time were the best we had in place. But they also strongly encouraged companies that as new methods became available, you're encouraged to move to those methods, again, to best control your product and residuals. So it carried on that way for quite some time. In 2004, there was a CMC strategy forum. There was a document published as a result of that in 2005, defining your product profile and maintaining control over it, part 2. It turns out this ended up being a very consequential paper for our industry in general, especially with respect to host cell protein. Some big things came from that. First of all, you should use a host cell protein ELISA. Some of these other methods are not really adequate to measure low levels of host cell protein in the presence of product. However, there was still some ideas of using 2D gels and SDS page to supplement the ELISA. So we hadn't got quite that far. It paved the way for using multi-product assays, which are essentially in-house platform assays. It also said if you have a well-developed generic or commercial assay, it should be appropriate for use. Again, if you can demonstrate that it's suitable for use. The big one, the big 2, is first of all, we all know that everybody goes by this number of 100 ppm acceptance criteria for host cell protein, and nobody knows where that came from. It actually came from this paper, the Champion paper of 2005. So that was it didn't mean to be, but it became quite an important number because we're still to this day, 20 years later, still kind of going to that number. But finally, it also is the first place where it said that you should use the host cell protein ELISA, but you absolutely should do coverage analysis to demonstrate that the antibodies in the assay are broadly reactive. And again, at that time, we were talking 2005, the only tool that we had available was 2D Western blot. All right, so that kind of paints a picture overall of kind of where we were. I'll talk a little bit about the ELISA and the Western blot, which are kind of the tools that we had available to us, and then show the consequences of that. So first we'll go into the immunoassay. This is a host cell protein ELISA in the format from Cygnus Technologies. So this is one of our commercial kits. You can see that the plates come pre-coated and blocked. There is a simultaneous incubation of the sample standards controls as well as the HRP labeled detection antibody. That's typically a 1 to 2 hour incubation depending on the assay that you're running. There is then a single wash step detection with TMB. So as you can see, while these are extremely complex assays to develop, we really go out of our way to make it as user-friendly as possible so that it can be successful in the hands of users of different levels of experience globally. So this is kind of a summary of the HCP ELISA. Again, as I showed before, there was a lot of methods initially used. The ELISA kind of made its way to the top as the gold standard for process monitoring as well as product lot release. The number one reason it can do this is it truly is the only tool that we have that you can measure nanogram per mil levels of host cell protein in the presence of mg per mil levels of product. It does not require a high level of expertise to run. However, as I said before, it does require a high level of expertise to develop one of these assays that is appropriate and broadly reactive. They tend to be quite robust and reliable, again, when developed well, which makes it very easily transferable. So whether that's to a QC lab down the hall or to a CDMO on the other side of the world, an assay like this is quite easy to manage, to qualify, and to validate. However, there are some drawbacks to the host cell protein ELISA. These need to be discussed because it really drives the importance of all the orthogonal methods supporting the assay. And the first one is the big one. a host cell protein assay always reports in mass units, typically nanogram per mil, and that's reported as nanogram per mg of product. However, that's not really the case, right? So these assays, we have no idea what host cell proteins are there or which ones the antibodies are reacting to. So what we do to make these assays is we have a total population of host cell protein, typically a harvest material, and we know the total concentration of host cell protein in there, not of the individuals. And our ELISA reports a signal based on the concentration of that standard. When we put our unknowns into the system, it gives us a signal. We equate that to our standard curve, and then that gives us the mass units. But as I said before, it doesn't tell you anything about what's there or what host cell proteins you're reactive to. The other thing is that these antibodies are polyclonal antibodies to thousands of individual host cell proteins. If you change your antibody, so if you extinguish your pool, or exhaust your pool, you have to make more, the chances are you're not going to make the exact same population of antibodies to the host cell protein, and the sample results will change. So it's really important that you make an assay that's appropriate and that you have a supply of those reagents to last for the long term. Because again, these antibodies can generally take about 12 months to generate, but another 6 to 12 months to develop, qualify, and validate your method. Okay, so the second tool we had available was the Western blot. So again, the champion paper did set a point that we needed to perform coverage. That was quickly picked up by regulatory agencies, and that was an expectation by about 2005. Again, we had a Western blot, not that it's the best tool, but it was the best tool that we had in 2005. So in this method, you run two, hopefully large format 2D SDS page gels. One you silver stain, and the second one is transferred to PVDF membrane and probed with the host cell protein antibodies. At the end of the experiment, you do spot counting, a lot of vectoring and other tricks, and you get some estimate of coverage. You can also look at quadrants of the gel to make sure you're not missing any particular type of host cell protein. So as I've said a couple of times now, again, Western blot's the tool we had, and we did it at Cygnus here for quite a long time, and we realized that the method has quite a few limitations. Limitations to the point where it made it very challenging to answer the questions that we were trying to answer, which is predominantly how will the antibodies perform in the ELISA, and is it truly broadly reactive? Some of these limitations are the sample preparation. So again, you're reducing these proteins and covering them with negative charges. Running gels, so you're doing typically 24 centimeter large format 2D SDS page gels. We're making some assumptions that if you load two individual gels, they'll run the same. That is rarely the case. Transferring host cell proteins to a PVDF membrane is as much an art as a science. I don't know how many of you had done that before, but if you transfer a gel for too long, all the small molecular weight proteins will blast through the membrane. But unfortunately, if you don't transfer for long enough, the large host cell proteins, large molecular weight, will never leave the gel. So it really is an art to it to optimize that transfer. And I think a lot of times when companies think that they're missing low molecular weight host cell proteins, that's typically the case if they're doing 2D Western blot. It's more of a transfer issue. Fixing gels is always interesting. Again, you take a 24 centimeter gel. When you do the fixing, it goes down to more of 11 or 12 centimeters. You have to rehydrate that gel and hope to get it to reasonably the exact same size as it started, so that it's comparable to the PVDF membrane. The last two are really important. The immunological challenges. So you are taking these three-dimensional proteins and linearizing them. So you have destroyed your conformational epitopes of these host cell proteins. So you're already limited in the epitopes available for detection, but also non-specificity. So in our experience, anytime you put a decent amount of any type of protein on a PVDF membrane, you will get an antibody binding to it. That's one of the challenges we have when people want to detect or test for cross-reactivity, because if you put more than 100 nanograms or so of a product protein on a Western blot, you'll get a good antibody response to that as well, even though it's nonspecific. So this is kind of where we were with the tools we had available to us at the time. There was a regulatory expectation that if you were starting out a process, preclinical to phase 2, it was perfectly acceptable to use a generic or a platform assay. However, there was also an expectation that if you move into phase 3 and beyond, that you would go to a process-specific assay. So again, it wasn't because we knew that the process-specific is better, because it's a topic for another day, but there was assumptions that if you made an assay that was specifically to your cell culture process, that it would probably be better than a generic or a platform. And again, that's all we really could do at the time, were make assumptions and do the best work we could, because we didn't have the tools available to us to know the answers to those questions. So next, let's step into some of the advances that we've made. Again, I'm just speaking to Cygnus Technologies today. So the first one is our antibody affinity extraction. We'll talk about the technology itself and why it's better than previous methods for coverage. And then we'll really dig deep into the mass spectrometry. So we'll look at it for coverage analysis. We'll look at it for coverage in product expressing, and probably what's becoming the most important and what the area that we're very excited about is identification and quantification of host cell protein and drug substance. Okay, so we'll start with antibody affinity extraction. So here we have our schematic of how this works. We start with a null or mock transfected harvest sample. We'll take some of that sample and set it aside. So you can see that's the pre-AAE aliquot. We then generate a column with the antibodies used in that host cell protein ELISA. We do extensive conditioning. We load the remainder of that sample on the column. We wash the column to baseline, and then we elute the bound host cell protein. We typically do this for 4 cycles. It's not to strip the sample, but it's to get enough host cell protein to perform our downstream analytics. So at the end of the 4 cycles, we pull them together, we concentrate and buffer exchange. And that leaves us with our 2 samples. So we have our pre-AAE aliquot and our post-AAE aliquot, which are all the host cell proteins that were specifically removed from that sample. And then for detection, so again, historically, we've been doing this since we developed it in 2013. We've done it commercially since 2014. So that started off doing large format 2D silver stain. So again, the immunological reactions happening in a much more native environment on that column. So detection can just be done with silver stain. The other detection method, which we're really happy with and moving most projects toward, is mass spectrometry. And there'll be quite a few slides under explaining why we're excited about that and the advantages that it presents over any gel-based methods. Okay, so here's raw data from our AAE with silver stain. This is our 3G CHO assay. You can see the pre-AAE is all host cell proteins in the harvest material on the left. The post-AAE, this is the elution fraction from the column. And I like to show this gel in raw format because, again, I like to reiterate that the point of doing coverage analysis is to demonstrate these antibodies are broadly reactive to the host cell proteins in your process. You really don't need to do the spot counting and the overlays here. It looks almost like identical images. When you look closely, obviously there are some differences, but the point is that you can see here they are broadly reactive, and that gets you a long way. However, they are gel-based methods, so they also have some limitations. And I think for this specific experiment, the coverage came out in the low to mid 70% range. So again, I said we'd talk about some of the advantages over previous methods, and the previous method is 2D Western blot. Again, it's a method that we've been running my entire career. But again, we realized that it really had a lot of limitations that made it not really answer the questions we were asking. We kind of went through those already, but AAE gets rid of most of them. And when you're doing silver stain gels, you still have the gels, so running the gels and fixing the gels are still there. But we eliminated most of them. And when we go into the mass spectrometry, we've eliminated all of them. But the number one question that we get for our AAE is specificity. So how do we know our column are not pulling down antibody or host cell proteins in a non-specific way? Well, first I'll just say that, I'll talk you through here. So on panel A, you can see this is our CHO host cell protein antigen passed over our CHO antibody. So you can see that there's a very high load peak. So we're loading high volumes of host cell protein. We wash the column to baseline. That's really important. And then we perform the elution. So you can kind of see that for each of the 4 cycles there. And you can see the nice elution peaks after we've washed to baseline. For the panel B, this is our CHO antigen passed over a non-immune goat IgG, so just normal goat IgG column. You see the same high loads. We wash to baseline, but there is no elution fraction. So this is just a good visual representation of the specificity of our AAE method. Well, I'll leave it at that. I won't mention that nobody ever does these negative control blots for 2D Western blot or asks about its specificity. Anyway. So now we'll go into mass spectrometry, and I really want to point to some of the real advantages of using mass spec. So first of all, you actually identify all the host cell proteins in the pre and the post. That's important for a couple of reasons. One is most of my career, we have companies come to us and ask, does your assay detect some particular host cell protein? And the answer has always been probably. But again, we didn't have the tools to really know the answer to that question. So here you can see on the left-hand side, which is PLBL2, we went and looked, and you can see for the pre, we could detect PLBL2, so we know it was there. But in the post, you can see it was greatly enriched. So by doing the 4 cycles of the AAE column, we're enriching it because we have antibodies specifically pulling it out of that sample. So this gives us some pretty strong data and indication that yes, our antibody does detect PLBL2. On the table on the right-hand side, this is kind of example data. And this is more than just example data because this is the high-risk host cell protein. So when you do gel-based methods, you're looking at spots and you're trying to line up the spots, and it really doesn't give you a lot of information other than these spots match or they don't. Here we're identifying all the host cell protein. And here you can see in the pre, for all of these problematic host cell proteins, if they're present or not. And then in the antibodies you're using as a test, so 550 and 550-1 in this case, you can see that the antibodies detected each of those problematic host cell proteins. So it gives you so much more information. Rather than just looking at spots, counting spots, you can say, these are the host cell proteins that are there. These are the problematic host cell proteins that are there. And, are we detecting them or not? Because these are the ones that are really important, the ones that can potentially cause problems, either their immunogenicity or problems with your product or its formulation. So it's really important information to have, and also makes you and the regulatory agency feel more comfortable in the assay that you've chosen. So here we have an example of the 2 side by side. So panel A is silver stain, AAE silver stain detection. You can see there's still a large number of spots. We're detecting well over 1,000 spots and have very high coverage. And here B, we're presenting the mass spec data slightly differently. We know everybody loves to see gel images. That's just a reality. But here again, you can get so much more information because we've identified them, but we also get the theoretical PI and molecular weight. And what we do to construct these graphs is that we can make matched spots appear green. So it's pre and post. Pre only means that our antibodies potentially have missed that protein. So you can look at those, the plot of those, and look at the distribution to see if there's a bias toward missing a particular type of protein. And finally, the post are the black spots. So the black spots are proteins that were probably below the detection limit in the pre-AAE, but we've enriched them through that process, and now we can detect them. So we would have never known they were there had we not done the AAE enrichment. And again, a higher number of spots in the pre and the post, and very high levels of coverage. Here's an example of 2D Western blot, AAE silver stain, and AAE mass spec. I just want to point out, a couple of things. First of all, Western blot, you're typically detecting a lot less spots. And again, due to all the limitations that I have stated before with transfer inefficiencies, with linear epitopes, things of that nature, it typically tends to underreport host cell protein, the coverage. And you can see here with the AAE silver stain and mass spec, again, we're getting much higher spot counts and very nice coverage. Here's an example of some of the other coverage that we've done. So we talk a lot about CHO because CHO is by far the most common expression platform used. But I just really wanted to point out here that Cygnus offers HCP analytics for pretty much every expression platform that's used across the industry. And we've done coverage analysis for most of them. And we have taken the time to build peptide libraries, again, across most of these expression platforms. So we can do our AAE mass spectrometry for at least all of these and a few more as well. Okay, so let's talk about identification of host cell protein in the drug substance. So I'll be first to point out that there is no regulatory expectation at this time to identify HCP in your drug substance. So they want you to do coverage analysis and then show them the qualification data in the ELISA. But I also really like to point out that regulatory expectations should be the minimum, right? So that's the bare minimum that you have to do to have your assay accepted. However, you should want to know more, right? So there's a lot of bad actor type proteins that can really cause problems with your success of your product down the road, right? So there's some that are just going to be immunogenic that could be problematic to your patients. But we're finding more and more some of these biologically active proteins, the lipases, the proteases, that can cause problems with the integrity of your product itself or with the formulation of your product. And it's really nice to be able to evaluate those early and not find out about them in the clinic or 3 to 6 months into a stability study. So yes, these are not regulatory expectations. However, it is in the company's best interest to fully understand the HCP and their drug substance in order to enable the best chance of success in the clinic. A quick schematic of how we do this experiment. Again, it's very similar to the original, except for you'll notice that the only option for detection is mass spectrometry. Again, that's because if there's product present, gel-based methods really become even less useful than they are without it. The product tends to be quite overwhelming, and you can't really get visibility into most host cell protein. That's kind of laid out here as well. We've always wanted to do mass spectrometry on drug substance, but the panel on the left kind of illustrates why we couldn't. You can see that there's a tremendously high level of product-related peptides here, so in this case, heavy chain and light chain, and you have very limited visibility into the host cell proteins that have persisted through the process. But with AAE, essentially acts as an affinity column for host cell protein, right? So you can deplete your product while enriching the individual host cell proteins. So for the first time, we really have great insight into all the host cell proteins that have persisted through the product, which means that we now have the ability to do a much more meaningful risk assessment to see how, what type of proteins are there. And we're working every day toward understanding how much of each of those are there as well. I'm not going to spend a ton of time here other than just to say that, AAE is the method that we use at Cygnus. It is common to do native digestion, which is a pretty common method used across the industry as well. We found that both work well, but in every case that the AAE is slightly better. You can typically get more host cell proteins and some that you typically would miss during native digestion. But it is something that, that we have thought about and worked through. If you'd like to talk more about it, we're happy to do that. Okay, so just to kind of wrap this up, AAE is much more predictive of antibody performance. So again, I've seen Western blot lead people down a lot of bad decision trees and into rabbit holes. Looking for cross-reactivity can be a problem, or thinking that you have much better coverage than you do, or thinking that you have very poor coverage even though the antibodies are great. So it really gets rid of a lot of those challenges. It is a great tool for any sample that contains drug substance. So sometimes you don't have access to mock or null transfected material. We can do AAE mass spec with harvest sample containing drug substance. That's not a problem. That's really not possible for gel-based methods. We can identify unique proteins. So again, it's lovely to see spots and get some assessment of coverage. But again, it's in our best interest to know what's there so that we can really mitigate any type of risk. So in knowing what's there and being able to track it through your process is a much better ideal situation. And then finally, it's the best tool for HCP identification in DS. It's the only tool that we've seen that really does well. And like I said, here at Cygnus, we've got identification covered pretty well, and we're working every day now towards quantification. So let's talk about some of that, some of the work that we're doing here. We have some relative quant methods that we're looking at, and that's going to help us with just looking at global relative levels of host cell protein. But as I've kind of mentioned earlier in the talk, there's absolutely some host cell proteins that we care more about than others. So, just in general, global levels, that's great. But if you have a protease or a lipase, you really want to know how much is there, because even very low levels could cause big problems down the road. So we've developed our Cygnus CHO Lipase Assay. So we have absolute quantification of these 6 CHO lipases using heavy labeled peptides. Again, I'm not going to go into great detail here. This is more just kind of an overview of the capability, and also to focus on the fact that we're expanding this library, and that if anybody is looking for specific host cell proteins, to again develop more methods, that we're obviously happy to do that. So let's just kind of go through a high-level review of this. So we accomplish this through parallel reaction monitoring. So it's targeted proteomic analysis through high-resolution mass spectrometry. It's very highly specific and sensitive, and I kind of hope to show you some of that data going forward. And our advantage that we like here is that we can do it on the same instrument that we do our global, our relative quant, our DDA and DIA-based methods. So here's kind of a general workflow. So we take our sample, we do our initial preparation, take it down to proteolysis to peptides. We spike in our stable isotopically labeled peptides at known concentrations, go into the LC-MS, data analysis, and then we have our protein identification and quantification. This is an example of kind of how we're doing the quantification. So again, we have known concentrations of the stable isotopically labeled peptides. We integrate that area under the curve, and we can translate that to the natural native peptides in the sample, the unknowns, and get absolute quantification there. Again, if you'd like to speak in more detail about this method and the capabilities in-house, you can reach out to our business development team or our technical services, and they're happy to to really get into real detail on these methods with you. So here we're looking at our isotopically labeled peptides versus non. You can see we perform standard curves and we have great linearity. So we feel really great with these methods. This kind of gives you the quantitative range for each of these lipases. You can see these are highly sensitive methods. So some of them, considerably less than 1 ppm. So again, compared to our relative quant, these are extremely sensitive methods. And that's really what you need, again, when we're talking about lipases and proteases, because an active lipase or protease, even at very low levels, can be a problem. Here we're looking at spiking our USP PLBL2 into drug substance samples, performing spike recovery at different levels. So you can see here that typically we're within 10 to 30% accuracy of spiking in the pure PLBL2 into a DS sample, even at very low levels. We'll probably skip over this one. Okay, and finally, this is kind of a real-world example of why does this matter? It is important, but we have a company that had come to us. They have 2 drug substances. They're different products from a different process, but they both shared a formulation buffer. And they noticed during their stability studies that the first one, the drug substance 1, had completely degraded in 3 months. The polysorbate had completely degraded in 3 months. The drug substance 2, again, a different process, but the same formulation buffer, had no measurable polysorbate degradation at the same time period. And they wanted us to help them understand what may be going on, right? So we looked at each of these samples in our 6 lipase assay. And you can see in drug substance 1, reasonable levels of lipases, most of these lipases in there, and that's contributed to the degradation of the polysorbate. In drug substance 2, 5 of the 6 were less than LOD, none detected, and PLBL2 was the only one found. So it's interesting in a couple of ways because, for the past several years, the industry has been chasing PLBL2 as the source of polysorbate degradation. I think we've all found that it was not. It was one that we could measure. It's these other lipases that are causing that. And this is just another example of that case. The presence of only PLBL2 did not degrade the polysorbate at all after 3 months of stability. Okay, so let's talk more about the ELISA. Specifically, decisions to be made when you're trying to select the right assay. So at the onset of this talk, we talked about, in the early days, kind of what were our options for assays to use. And again, we didn't have a lot of tools in our tool bag to know that we were using the right assay. I hope that I've at least done some work to show you that we do now. We can tell you what host cell proteins are there, which ones we're reacting to, or even getting really close to tell you how much. But the big take home from that is that these bottom 2 avenues are now available as well. So you can still use your generic or platform assay, but going into phase 3, you don't have to do a process-specific assay. If you can put together the analytical package that justifies the generic or the platform method, that is an acceptable path. But again, just like the process-specific, you have to do the work to show that that assay is going to be acceptable and adequate for that process. So how do we start when we're trying to choose our ELISA? So clearly, we're scientists. That's where we always start, is based on the science. You want to find an assay that is going to give you the best understanding of your host cell protein that is in your process as well as carrying through to your drug substance. So you can use some of these methods like the coverage analysis, identification in the drug substance, all that type of stuff, to make sure that from a scientific perspective that you're using the ELISA that gives you the best understanding and the best reading of the host cell proteins that are in your process. So a couple that are a little less thought about are regulatory considerations. So there is just a reality that if you decide to go with your platform method or a commercial assay, generally speaking, you're going to have more questions. Again, that's just still based on some of the assumptions that we made in the early days that a process specific is by default better. I don't think that was completely thought through, because who developed that process specific assay is really important. But just understand that you will have more questions and you'll need to put together probably a more extensive package. And then harmonization of regulatory expectations. So globally, again, in early days, it was kind of frustrating because depending on what country you were filing in, there could be very different expectations. I will admit that now that has actually gone quite well, and there's pretty good harmonization across countries as far as their general expectations. However, that's not always the case within an agency. So different reviewers have different levels of experience with host cell protein review, and you can get quite varied amount of or types of or in-depth questions depending on the reviewer that you have for your product. Okay, so once you've made that decision, how do you know that the assay that you've selected is appropriate? The big take home here, most customers come to us and they say, I have an assay, the coverage is 85, 70%. Is that good enough? And my answer is, I have no idea, because you cannot decide if an assay is adequate or fit for purpose based specifically on coverage. That is only one part of determining if the assay is appropriate. Coverage analysis only answers the question, is the antibody broadly reactive to the host cell proteins in your process? You still need to do the work in the ELISA, looking at dilution linearity, looking at accuracy, precision, as well as the range of the method, to get a full picture and present a full package of if the assay is appropriate for use. So just keep in mind that coverage analysis is only one part of that question, and that it should not be looked at in a vacuum. It's part of the total package. Dilution linearity, I personally find to be probably one of the most important things that you can do. If you look at the table, it's just kind of an illustration of what that means. So you can kind of see, at first at 1 to 2 to 1 to 4, when you multiply the result by the dilution factor, the results tend to be increasing. However, from 1 to 8 on, as you multiply the result by the dilution factor, you get relatively the same result. When you can show this for all of your in-process samples and hopefully your drug substance, that means that you have antibody excess for all of the host cell proteins that are in that process. That gives you a pretty good feeling that the assay is probably going to be okay. If during your process you lose dilutional linearity, that's also a red flag that your purification process might have a problem, and you could be enriching host cell protein. So this is typically where you start. This is the first thing you do when you're assessing the suitability of a new host cell protein ELISA. Look at dilutional linearity for your in-process and DS samples. And if you can show linearity, then you can feel pretty comfortable that it's going to be okay, even though you need to do everything. Coverage analysis, again, we highly recommend AAE mass spectrometry, again, because we're trying to put together a compelling package. And yes, you can look at spots and you can get some idea of coverage, but when you can identify them, you can still get your coverage. But in addition to that, you can say, I have high coverage. And by the way, these are all the problematic host cell proteins that I found in my process, and the antibody recognizes all of them. So showing the high coverage, but additionally demonstrating that you have coverage to all the problematic host cell proteins is a much more compelling argument or case to be made if you're trying to convince yourself or a regulatory agency that the assay is appropriate for use. Here we're looking at if you have two particular assays, whether you're assessing the commercial assay versus a custom or process specific, or if you're even bridging from one set of reagents to another. Here's kind of an example of how you can approach that. So here, this is just an example of our bridging from F550 to F550-1, and you can see we did coverage analysis to the same antigen. Both of them recognized basically the same number of proteins in pre and post. Both of them had very high coverage analysis. So that made us feel pretty comfortable that we were resupplying this assay with the antibodies that were going to be very similar. But in addition to coverage, the next logical question is, okay, they have high coverage, but are they detecting the same thing? So here we put it into a Venn diagram, overlaying the different host cell proteins identified by each antibody pool. And you can see we had a 96% similarity between the two antibodies. So again, in this situation, if you have these two antibodies or two assays that you're trying to decide between, the coverage looks great. You can see that they're both measuring the same thing. Then you can start entering into some of these business decisions, that we talk about as far as, supply and control and those type of things. Okay, so this is just phase-appropriate HCP analysis. Again, as we discussed before, early on, generic assays, platform assays, they're great. They're accepted by all regulatory agencies. As you move to phase 3, thanks to a lot of these tools that we now have at our disposal, you can stay with the generic or commercial kit, your platform kit, or you can develop a process-specific kit. And if you have any questions as far as why you would use one of those versus another, we're happy to have those conversations with you to kind of help guide you through that process. Services, Cygnus offers all of the services that we've talked about in this webinar and a lot more. But you can kind of see in the bottom here, coverage analysis, identification of HCP and the drug substance, any of these types of services we're happy to assist you with. Don't have time due to the time constraints today. We don't have time to go through all of these, but again, if you'd like to discuss any of this, you can reach out again to our business development team as well as our technical services team. We're happy to have those conversations. Okay, so finally, make sure you're using the best assay for process development, product release. And the only way to do that is by using the best tools available. You want to make sure that you're using the best tool that you can to fully control and understand the HCP in your process. And if you're looking at and counting spots on a gel, it really doesn't give you that information to really answer those questions. You can get an idea of coverage, but as far as what's actually there, what's making it through your process, all of those types of questions can only be answered using advanced orthogonal methods, specifically mass spectrometry. Make science-based decisions when choosing your assay. So again, we're trying to really move away from some of these academic assumptions that we had to make in the early days. A lot of companies and regulatory agencies are still kind of there, but we do have the tools to know if an assay is appropriate. So you should use those at your disposal when you're trying to decide which assay to use. And then finally, to wrap up, if you're going to use a generic or commercial assay for product lot release, make sure you stay in close communications with Cygnus Technologies. We're here to be your partner to help you through that process. And also we can work with you as far as control and providing long-term support for that method. Okay, so finally, a lot of the people on this end that kind of helped put this together, a lot of the work and the presentation itself. Dr. Jared Isaac, he runs our chromatography mass spectrometry group. Ala Zilberman runs, she's a VP of Business Development and Technical Marketing, and John Locklear runs our Assay Development and Technical Services group. So with that, I think we have a little bit of time left so we can entertain some questions.
Yes, absolutely. Thank you, Eric, for such a great presentation. And for our audience here, I would like to remind you how to ask questions. You can do so by submitting them in the Q&A box, which can be found directly below the video player. So the first question I have for you here, Eric, it says, has generic reactive MABs on the AAE column provide the same coverage as product-specific reactive MABs?
So we're going to have to step back from that. These are all polyclonal antibodies. And yeah, I mean, so how they react on an AAE is really more of how they were developed in the first place, right? So there is polyclonal antibodies for host cell protein are not your typical immunization, right? So there's very specific ways that we've worked out to make sure you're making antibodies that are broadly reactive to all of the host cell protein. If you lack that expertise and do traditional polyclonal antibody generation, you will miss a significant number of those host cell proteins, and therefore you will not have the same coverage by AAE. So the AAE is really just the downstream part. It's like the computer, it's the garbage in, garbage out, right? So if you make great antibodies, you'll get good results on AAE. If you don't, then the results will suffer.
All right, great. Thank you. Our next question here from the audience says, has coverage analysis by AAE with the 2D page silver stain or PRM-MS been submitted to and accepted by regulatory agencies?
Absolutely. So again, we have offered AAE as a service since 2014. We've done over 500 service projects with silver stain detection as well as mass spectrometry detection. It has been accepted globally, regulatory agencies around the world, and has also been written into Chapter 1132 of USP as an acceptable method of coverage. In that document, it's known as immunoaffinity chromatography.
Great, thank you. We have another question here about the columns, which are they available for purchase at this time?
They are not. So AAE is a proprietary service that we offer at Cygnus, but we are happy to partner with you and perform the coverage analysis for you.
Great, thank you. We have a few questions here about the coverage percentage. So the first one here is, what is the acceptable percentage HCP antibody coverage?
Yeah, that's the $1 million question. No, there really is no specific number that is acceptable for coverage, right? So regulatory agencies will not give you a number, and part of that, it's not them just being coy. There are reasons for that. There are scenarios that roll in, like the expression platform that you're using. For example, CHO, we have a lot of safety data for that. It's the dose, frequency, and route of administration for the drug that you're going to be using. There's a lot of things that come into play with what is going to be acceptable or not. And also, as I demonstrated before, the method that you use could be just as predictive of the number you're going to get as the quality of the reagents. So again, we're just trying to demonstrate that the antibodies are broadly reactive, and I would be a lot less concerned about the number than demonstrating broad reactivity, and then looking at the performance in the ELISA and take that entire package as an assessment of the quality of the assay.
Okay, great. Thank you. And regarding the AAE method that you've talked about, does it overestimate HCP antibody coverage percentage?
Right. Yeah. So again, that goes back to the specificity that I was talking about earlier in the talk. We take a lot of great pains to make sure that we do have a specific method, and that we're not pulling host cell proteins in a non-specific way. We have done that, from the beginning to address that. But I think it's often thought that we do, because again, Western blot tends to really under-report coverage. Again, due to some of the immunological challenges that it has, as well as transfer inefficiencies, and sample preparation. People are typically used to seeing these lower numbers from 2D Western blot, and then when you put out a method, regardless of how well you've shown that it's specific, as well as supporting data, the numbers tend to be higher and looked at somewhat skeptically. But no, it does not.
Fantastic. Well, with that, we will wrap up today's event. I want to thank the audience for attending and for participating in today's event. I would also like to thank Eric for such a great presentation today, and our sponsor, Cygnus Technologies, for making today's webcast possible. We would like to ask everyone in the audience to participate in a brief survey. This survey will appear on your screen after today's presentation is ended. You'll receive an email alerting you when this webcast will be available for replay, and we invite you to forward that to your colleagues who may have missed today's live event. Thank you everyone for joining, and we'll see you next time. Have a great rest of your day.